Yeah, you able to see my screen now? Yeah, okay. So Jaya Patel, you were mentioning, sorry for the interruption. Yeah, so you were
Okay.
mentioning about eucalyptol and how, okay, we were looking at the structure, if any of these were phenolics. Any idea about that?
Vanillin. Okay, vanillin is phenolic aldehyde.
Huh.
Okay.
And
And
others?
Epileptic is having a lol. That means it contains the alcohol group. OH. So it is also phenolic.
No, OH doesn't mean phenolic. OH means it's alcoholic.
Benzene with OH. That is known as a
Benzene with,
phenolic
yeah,
alcohol.
yeah.
Okay. Yeah. But right now, sir, I am not recognizing all the structures.
Okay. What you mentioned is right here. In this question, it's vanillin, and the only difference is it doesn't have a phenol. Also, it is also cyclic. There is a ring
Hmm.
present in eucalyptol. It is not a phenol per se, and that's why we go with vanillin for this question. Right,
Okay.
right. Um, yeah, good. We'll go to the next question. So the next question is match the plant source on the left hand side with the appropriate active compound on the right hand side. So you have opium poppy and chinchona tree, and on the right hand side you have taxol, quinine, and morphine. So this could be a simple, straightforward question to answer. You can take a couple of minutes, and then people can start raising your
Sir,
hands.
answer is C. Answer is C.
Right. One.
Often poppy contain morphine and cinchona contain quinine.
B and B2.
Right, one minute. If you know the answer, you can raise your hand. We can take turns because there are a few people, about 15 people who joined. So yeah, yeah. Since you answered the last question, Jaya, we'll go with Nisha. Yeah, Nisha, you can start by mentioning the answer.
Yeah. Good evening, sir.
Good evening.
The answer is C.
Okay. Uh, yeah. Do you want to discuss about this? Like, do you know something more?
Something more like,
What is morphine? What is quinine? And what is Taxol?
okay, these are actually alkaloids, sir.
Okay, that's the based on the chemistry you are saying. Okay.
Yeah,
So do you know where morphine is used routinely and where quinine is used routinely?
pardon. Actually, pardon?
Do you know what is the purpose of morphine and quinine?
Analgesic. Morphine is analgesic.
Right. It reduces
That is
pain. Huh.
yeah, quinine is anti-malarial,
Yeah. And Taxol? Does anyone?
anti-cancerous, yeah.
And where do we get Taxol from?
Taxus brevifolia.
Right. Okay. So people are aware of this. Good. Yeah. Um, hmm. Yeah, you can mute your mic. We'll go to the next question.
Oh,
Yeah. The third question is which of the following compounds serves as both primary and specialized, that is secondary metabolite? And four options are given here: glucose, cannavanin, deoxyribose, and anthocyanin. So before anyone unmutes, what I would suggest is if you know, you can raise your hand. And to start with, one can mention what a primary and a secondary metabolite is. And from that, we'll take it over to reach the right answer. You can take a couple of minutes, and whoever knows the right answer can raise your hand. Yeah, Nisha, you can start by first mentioning what primary metabolites are and what secondary metabolites are. And then we can discuss and then reach the right answer.
So primary metabolites, these are the metabolites from which the secondary metabolites are formed. Like you have amino acids, carbohydrates, proteins. So they, yeah, through certain metabolic pathways, these secondary metabolites are produced.
Okay, but why are primary metabolites called primary? Any idea? Based on their functionality or when and how much they're produced? Do you have an idea about primary metabolites that way?
Yeah, actually, yes, I have the idea. Mm,
So, primary metabolites are okay. Before we go, does anyone else want to try? The question that I wanted you to answer is what is the difference between primary metabolites and secondary metabolites? Yeah, Jay, you can unmute.
So primary metabolite are those which are found inside the body, and secondary metabolite are those which are
Those which are
those which are from outer side.
need
Outside.
not
Need not
be the
be
case.
the case.
So
So
you can mute.
you
I
can
think there's
mute.
an
I
echo.
think there's an echo.
Yeah. So primary metabolites. So primary metabolites are those that are constitutively produced, like independent of a particular growth stage, because it is essential for the survival of the cell. But whereas secondary metabolites are specific to a particular function. Like let's say in a particular growth stage, a metabolite is needed for expressing a particular function. Uh, like something to do with reproduction of plants. So the plant may be requiring to synthesize a volatile compound to attract pollinators. So those chemicals at those particular stages are produced, and these are secondary metabolites. And this exists across the spectra. Now, coming back to the question that we have here, which of these are primary metabolites? Only primary, not secondary. Yeah, Nisha.
I think it's glucose. It's a carbohydrate.
And what about deoxyribose? Is it
That is also,
familiar?
yeah.
Is it familiar? Like, where is
Yes,
deoxyribose needed?
that I don't remember.
Okay, so deoxyribose is your component in DNA. So
Yeah.
deoxyribonucleic acids. So without deoxyribose, you will not have DNA, which means there will be no replication. And replication is essential for the cell to survive. So deoxyribose is also a primary metabolite. Now, with respect to the question here, uh, you- Nisha, what were you mentioning as the right answer?
So cannavanine, I think so.
Okay. Do you know about cannabinoid?
So actually by name it's looking like it's a amino acid and it is found in some plant also. Like
Okay. Okay. Uh,
I'm guessing it.
okay. We'll check with Jaya and Ajay. Oh, yeah. Jaya, you can unmute.
The right answer is anthocyanin. That is
Why?
give a color to the different fruits and vegetable.
Sorry?
That is present in the fruits and flowers.
But
Anthocyanin. That is a pigment.
is it a primary metabolite? Is it always produced?
Uh, no, it is a secondary metabolite.
Okay.
Anthocyanin is.
Okay. So the question that we are
And
dealing
the
with,
cannabidiol is the amino acids.
right,
So amino acids is a primary metabolites.
right.
Yeah.
And so we're not sure, is it? Whether it's canavanine or anthocyanin.
No, so it is anthocyanin, sir.
But the question asks that this compound can serve as both a primary and a
Both.
specialized metabolite. So do you think anthocyanin would be the right answer?
Okay, so cannabinoid, huh? Yeah.
Okay,
Both is cannabinoid. Yeah, yeah.
okay. Let's listen to Ajay's answer. Yeah, Ajay, you can unmute Ajay. Yeah, Ajay,
Yeah, Jay,
you
you,
can tell
you
us.
can tell us.
No, no, sir. Actually, miss.
Okay, so the answer to this question is canavanine. So if you, I hope you would have learned about amino acids. So canavanine is an amino acid that is very similar to arginine. So the only difference structurally is that there is an O group here instead of a CH2, which means that it's a it's an amino acid. And the role that this plays is that it can code for defenses against natural enemies. So what is studied is that when herbivores try to feed on plants, this canavenin would have been incorporated into a particular uh, protein as an amino acid instead of arginine. So what happens is that these insects will not be able to metabolize canavenin like it could do for arginine, because of which the insects would suffer and it acts as a defense mechanism. That is how canavenin acts as a secondary metabolite. So, and in some cases, it has also been observed that cannabinoid can be used for storage of nitrogen because it has these amino groups here. So that is how it can act as a primary metabolite as well. So this is why cannabinoid is the right answer for this question. I hope that was clear. If there are any doubts, you can unmute and ask me. Okay. If there are no doubts, we'll go to the next question. So the next question is, totipotency is a feature unique to the plant kingdom. Totipotency can be defined as, and four different options are given, a process through which terminally differentiated cells regain their ability to divide or become less differentiated. Option B is the ability of a single cell to divide and produce fertile adult individual. Option C is the ability of de-differentiated cells to divide and attain roles of specialized terminally differentiated cells. And option D, it's a process through which, from a mature organ under certain conditions of growth conditions, a lump of undifferentiated cells is produced. So, whoever knows the answer can wait for a couple of minutes and then start raising your hand. We'll discuss what the right answer is. We'll discuss what each of these terms is called as, and then reach the right answer. So, we'll take a couple of minutes and then you can start raising your hands. Yes, Swati. We'll take a couple of minutes and then you can unmute yourself. Yeah, Swati can go first, then Anvay can go. Swati, you can start by eliminating an option by telling us what a particular option here is referred to as. You can unmute Swati.
I think it is option B, ability of a single cell to divide and produce fertile adult individual, totipotency.
Okay, are you aware of the terms that are associated with other options? We can look at them as well so that we know we can relate to totipotency that way. So, do you know of any other option?
Yeah. First, I thought it can be the
Hmm.
third one,
Okay.
but yeah, but I think totipotency is the ability of the single cell to divide. Uh,
Okay. Um, Anvedana, do you want to try and describe what the other options could possibly mean? Like, for example, does option A have a particular term?
No, sir. I also tell the option B.
Okay, fine. So, I have posted an image here. So, what one needs to know is that there are a few technical terms that people use with respect to the ability of a cell to grow into a particular tissue and then going back to that original state and then growing into a new tissue. So, the first thing that we need to know is that when a particular cell, which can possibly grow into different tissues, if it chooses to take a particular route to grow into a particular tissue, that is called differentiation. And now, if this differentiated cell, under the stimulus of particular chemicals, if it goes back to its original state from which it had differentiated, that is called de-differentiation. So now you have the original cell itself. And now if this particular original cell is able to grow into a different tissue compared to what it was earlier, this is called redifferentiation. So, so these are a few things that you need to know with respect to plant cell growth. The next question is, in plant tissue cultures, sometimes elicitors are used to upregulate the accumulation of desired special metabolized. For a molecule to act as an elicitor, it must option A get recognized by a membrane bound cell surface receptor, option B be able to act as a transcription factor, option C have a nuclear localization signal, and option D be more than 10 kilodalton in size. So we can take a couple of minutes and then start by describing what an elicitor is and what it would, and the way in which it would give rise to a phenotypic or genotypic effect. So whoever knows the answer can raise your hand up after a couple of minutes. Yeah, Swati, you can unmute and you can tell us, you can start by telling us what an elicitor is. Swati, can you hear me?
Yeah.
Yeah, so because you were raising your hand. Yeah, so,
No, no, sorry,
oh,
sorry. It was before only it was like that.
okay, fine. Do you want to try answering this question?
No, I'm not getting the question only. Sorry.
Okay, do you know what an elicitor is with respect to plant tissue culture?
Elicitor which acts as a defense for the plant, defense system for the plant.
As in, like, is it produced by the plant or is it coming from outside? Any idea?
Mm, actually, today morning I heard, means I have taken the notes. Now I'm not recollecting.
Okay, so we can discuss this. It'll be easier. So elicit means it has to give a response. So like you mentioned, defense could be one of the responses. So let's say there is a molecule in a plant. Now this molecule is supposed to trigger a particular reaction. So the question that they're asking is, what are the features that this elicitor should have? So if the molecule should trigger a particular phenomenon, what all do you think are the required features? We'll start with a few options that are given here. The first option that we'll deal with is option D. Do you think that the molecule should be small or large for it to elicit a particular response? Okay, so one simple way to understand this is if molecules are small, they can diffuse easily, which means they can travel faster, and they would also have specific binding partners where they can bind to easily compared to large molecules. 10 kDa means 10,000 Daltons. That is equivalent to having about 100 amino acids, a small protein. It will be difficult to transport to first of all, synthesize this protein and then transport this protein within a plant. So, elicitors are supposed to be small. So, we can eliminate option D. The second option is it should have a nuclear localization signal. So, does anyone know what a nuclear localization signal is? Whoever knows what a nuclear localization signal is can unmute. Okay, so nuclear localization signal is a stretch of amino acid residues that would allow a protein to enter the nucleus of a eukaryotic system. But since we are dealing with small molecules, they can diffuse easily, and in through non-specific transporters, they can diffuse. So there is no issue with the presence of nuclear localization signal. So we can eliminate that option as well. And the option B is, it states that it should be able to act as a transcription factor. So does anyone here know what transcription factors are? Dipali, Anve, Hirvita, Maitri. You can unmute if you know what a transcription factor is. Others can also try. Pratiksha, Ritika, Sakshi, and others. Yeah, Nisha, do you know what a transcription factor is?
Transcription, it is a process from where we can synthesize mRNA through DNA.
Huh.
And I think factors, there must be some enzymes or some like which help to in the formation of mRNA from DNA.
Right, partially right. So transcription factors are not enzymes, but they are structural proteins. So they recognize something called as the Pribnow box. Do you know the -10 and -35 regions in a DNA? The promoter region?
The promoter region is there, yes.
Yeah. So transcription factors either sit on this -10 and -35 region, allowing RNA polymerase to synthesize mRNA, or they bind to other regions. Anyways, they help the RNA polymerase and to create the mRNA. So you are right about that. But since these small molecules are smaller than DNA, they will not be able to sit on that. So option B, which is be able to act as transcription factor, that is also wrong. So what an elicitor should be capable of doing is that it should be able to bind to specific receptors, and specific receptors will then transduce this binding as a conformational change, and then it will pass down this information throughout the cell. And once this information is passed down throughout the cell, genetically a signal goes such that upregulation for specialized metabolites is passed on. So then these enzymes are synthesized and the specialized metabolites in the pathway are created. So for a molecule to act as an elicitor, the only requirement is that it should be able to bind to a specific receptor. Was that clear? Okay, we'll go to the next question. The next question is, identify the compound from the following that is of secondary origin but attain primary function. So there are four options given here: cellulose, lignin, linalool, and rosmarinic acid. So you can take a couple of minutes and then raise your hands if you know the answer to this question. Yeah, Anbej, you can unmute yourself. So we'll start with wrong options. You can justify why an option is wrong before we go to the right
So
option. Yeah.
the right answer will be lignin because cellulose is the primary and lignin is secondary. But lignin also function as primary as well for rigidity and stability for a plant.
Where is lignin present?
Cell wall.
Hmm. Right, right. You can answer it confidently. Yeah. And what about uh, cellulose?
Cellulose also present in cell wall.
What is
The most
cellulose?
most what is cellulose?
Yeah.
Uh, a component which is responsible for structural support.
Uh,
Chemically, cellulose is a carbohydrate.
okay. So are carbohydrates structural or do they perform some other function?
Mm.
Can cells break down cellulose to consume to use it as a carbohydrate? Is it like starch? Does anyone have an idea about that? Yeah, Ajay.
As a reserve energy.
Hmm. So you are saying it could be used as a energy storage. Hmm.
Hmm.
Okay. So here, how in order to understand this particular question and answer that, you need to know the structure of these compounds. So if you look at the structure of lignin, okay, we can first we can eliminate linalool and rosmarinic acid because these are not of primary function. But when you go to cellulose and lignin, there could be this doubt that okay, these are present in cell wall, but are they were they a part of secondary origin or something of that sort? So, firstly, like Deepali mentioned, cellulose is a carbohydrate. It's a polymer of the carbohydrate, and carbohydrates were always of primary origin. There was no secondary origin. So, with that, we can eliminate cellulose. And with respect to lignin, if you look at the structure of lignin, it is formed by oxidative coupling of few alcohols, which is comaryl alcohol, coniferyl alcohol, and synapyl alcohol. So it is a phenylpropanoid polymer, and this is how the structure of it looks like. And in the class, in the lecture videos, the professor had mentioned how through evolution there was this ability of plants to grow from just water to be able to grow to land and then adapt to land. So it was through this process that lignin became important and enzymes started acting on lignin like molecules or the precursors of lignin to be able to give it a role in such a way that it can adapt to land. So that is what we need to know with respect to the origin of lignin from secondary origin, but finally attaining a primary function. So in the initial years, it was not very important because the environment in which plant grew was not the soil. It was not a land plant back then. But once land plants started growing, what one could observe is that lignin was essential and hence it became primary. The next question is identify from the following which is not a suitable plant cell and organ culture technique for production of specialized metabolites. Option A, multiple shoot culture. Option B, adventitious root culture. Option C, cell suspension culture. And option D, somatic embryogenesis. So, whoever knows the answer to this question can first mention about the incorrect options and where or how it is used for what purpose. And then we'll go to the right option. So Anvejana and Ajay Tomar have raised their hands. Uh, yeah, one of you can unmute in a couple of minutes and start answering. Okay, does anyone want to try? Nisha, Raven, and Deepali. You can try attempting. We can discuss and we'll get to know, we'll get to learn.
Sir,
Yeah,
I think the answer for this is C, cell suspension culture,
wow, okay.
because it is a method to
Hmm.
grow cells in a medium. And others, when we talk about like multiple shoot cultures, adventitious root culture, and somatic embryogenesis,
Hmm.
they all are used for, yes, they all can be used for the new plants. I mean to say for the PTCs,
Plant tissue
I
culture.
think.
Okay.
Yeah.
So the question here is for specialized metabolites. So do you think cell suspension culture is not suitable for specialized metabolites?
Um, yes, I think so, but not sure for the answer.
Hmm.
Uh, because here only the medium is being used for when we talk about the technique of the culture medium. So in that sense, if I think cell suspension is not.
No, the question is when you do cell suspension culture, do you get specialized
Hmm.
metabolites? Can you get specialized metabolites?
So it's a method to grow these cells, right? Growing of
Yes,
cells.
yes.
Uh,
So now in cell
so,
suspension culture, will you get tissues?
hmm.
Okay, if you're getting tissues, can they produce specialized metabolites?
Yes, if we think with that inclination,
Hmm.
then yes.
Okay. Okay. So cell suspension culture is not the right answer. Do we agree?
Yes, yes, understood.
Okay. Okay. Okay.
So I think B is right answer.
Adventitious root culture. You,
Yes,
yeah,
sir.
yeah, Ajay, you can tell us what adventitious root culture is. One
Sir,
minute, Anvay. One
So I
minute.
think it's the A is the right answer.
Wait. 1 minute, Anvay. And Sushal and Swati, give me a minute. We'll go to your answer as well. Uh, yeah, Ajay. So you were saying adventitious root culture. You can tell us what
so
adventitious root culture is,
so because
whatever you know.
accept B, all three are able to grow like multiple shoot culture in rows. Can we see?
Can you repeat? Sorry.
So जैसे multiple shoot culture when we see
Huh.
in rows
Huh.
or somatic embryogenesis seen in vitro culture,
Hmm.
but in case of adventitious root culture, I
Okay, that was not very clear to me. We'll listen to what others have to say. Yeah, Sushal.
Hello, sir.
Yeah, you
I
can.
would tell the
Huh.
answer is somatic embryogenesis.
Okay, but before we go to that, we wanted to look at why other options could be incorrect. So, do you want to share with us what you know about other options like adventitious root culture, or
So I should tell why is that a viable option to produce the specialized metabolites?
you can first tell us what adventitious root culture is. So we'll take it from there. It will be a discussion session. Yeah.
Okay, so with what I know, so adventitious root culture is something where you produce like a mother part in the culture media, which will then produce multiple roots in it, like
Okay.
and like form the secondary root inside the culture media.
Hmm. So would that mean that it can produce specialized metabolites?
Yeah, I think if the metabolites, particularly say some metabolite that's produced in roots of a plant,
Hmm.
so you'll be able to produce it when you do adventitious root culture, right?
Right. So you have specific metabolites that are needed to grow the roots or proliferate the roots that
Yeah,
need not be primary for like throughout for all stages of the plants, but specifically at the root stage growth, because of which adventitious root culture can produce specialized metabolites. That's what you're trying to say, right?
yeah.
Right. And do you know about multiple shoot culture?
So again, it's the same. You put a tip meristem of a plant, and then you have shoot cultures of the same plant that can give rise to a uh, say more number of shoots of the same plant in the culture.
So the only difference is your starting material. What is the
Yeah,
source of your
it's
cells? Right.
yeah.
Right. Okay. Thanks. Uh, Yeah, I think it was Swati and Anvay. Yeah, Swati, you can tell us what you know about adventitious root culture and multiple shoot culture.
I think the answer can be A, multiple
Okay.
shoot culture, but I'm not sure.
Do you think in shoots specialized metabolites can be produced?
Mm, yes.
Okay, so that means it is a suitable method, right? The question is, We wanted to know which is
Okay.
not a suitable method. So that way we'll have to eliminate multiple shoot culture.
Yeah.
Okay. And Anvay?
Sir, I think the option A should be correct because in multiple shoot culture they are primarily focused on making large number of shoots from a single plant,
Yeah,
and the
but in
other three
that
options
case,
they are
do,
slightly
yeah,
different from this.
yeah. But the question is, when you think of these questions, you need to read these carefully. So they ask whether it can be used for making specialized metabolites. And do you think specialized metabolites are produced in the shoot?
Uh, I don't know, sir.
Okay, so one way to think of this is when you have a differentiated cell, it will have specific specialized metabolites. And as long as you use a differentiated cell for your tissue culture as a starting material, of course, you will have these specialized metabolites that are being produced in the tissue. This is where somatic embryogenesis differs. So you have a somatic cell and then a somatic embryo. And this could give rise to your seedling. And here you don't have specialized metabolites, but only the primary metabolites that are needed for its growth. The next question is, which among the following can potentially be used as an elicitor for secondary metabolite induction in plants? So for this question, again, we start with discussing what are all the incorrect options and why it is incorrect. Okay. Okay. So to answer Tanmoy's question, um, yeah, Swati, you can start by telling what is an elicitor. We have discussed that today in the class.
Yeah. Yeah, it can produce a secondary metabolite induction in the plant and act as a defense. So I think jasmonic acid can be the answer.
Okay, but before we go to
Increase
that,
the
what is
chemical as a
an
secondary
elicitor?
metabolite.
What is an elicitor?
It can increase the secondary metabolite induction.
Okay. Do you want to describe a bit more about that?
Sir, it triggers the production of secondary metabolites, sir.
Okay. So when will it trigger the production of secondary metabolite?
Mm,
When it will be attacked by some herbivores, something like that.
Yeah, based on environmental
yes.
cues. So,
Yeah.
yeah.
Environmental factors.
Huh. So, elicitors are the small molecules. So, what these small molecules do is, in response to a particular environmental stimuli, these would go and allow for expression of certain genes. And these certain genes are responsible for your secondary metabolite production. And these secondary metabolites act in response to that environmental stimuli. So, could you get that, Tanmoy? Okay, good. Huh. So, now we look at what properties, based on the options given here, we can discuss what properties can confer or allow a molecule to be an elicitor. So, Ravisha, do you think any of these options are incorrect, or do you think all these options can be possible elicitors? Ravisha?
Sir, jasmonic acid, sir.
Yeah, that is a elicitor. But what about other options? Do you want to comment on them?
Uh, Sir, I have the idea about the
Glyphosate?
option uh, glyphosate, sir. So it is an
Okay,
herbicide, sir. It is an herbicide.
okay. Hmm.
Uh, and maybe regarding with hydrocyanic acid also will be the same, sir.
Is that also a herbicide?
Yeah.
Okay. And kanamycin?
Kanamycin. Kanamycin.
Kanamycin.
It's an antibiotic. Right.
Yeah, yes,
So,
sir.
right. So, the reason for application of these, like for example, kanamycin or glyphosate or hydrocyanic acid, is that
Mm,
they would prevent pests or herbicides to affect your plants. So they do not elicit a response from the plants, but rather
yes.
they would directly act on the pests that are trying to encroach the plants. Whereas jasmonic acid, it is an elicitor for secondary metabolite induction. And this is how jasmonic acid looks like. So, and it has various functions that has been mentioned here. The next question is, which among the following is not a strategy followed by orchids for reproductive purposes? So the question is not a strategy. Option A, emission of phytoalexins to attract pollinators. Option B, flower mimicking the pollinator mate. Option C, emission of sex pheromones mimicking compounds of its pollinators. And option D, emission of pollinator repelling compounds post pollination. So, whoever would like to answer can take a couple of minutes and start with mentioning why an option could be an incorrect option. Okay. Does anyone want to try this? So you just need to mention why a particular option could be incorrect.
So I'll shall I tell, sir?
Yeah, yeah. You can start with one option. Why you think a particular option could be incorrect.
Okay, so I can go with C. So emission of sex pheromones mimicking compounds of its pollinator.
Right. Why do you think
So
so?
because like you have seen in many orchids where uh, it mimics to be a female bee kind of, so where the
Hmm.
male comes and copulates with it, so and that is how the pollination happens.
Right.
But if so many factors play a role there. So one factor is production of sex pheromones from that flower,
Hmm.
so that it will so the pollinators get attracted to the sex pheromone and go and copulate with the flower.
So do you know what pheromones are?
Yeah, so they are compounds that attract like the opposite sexes for mating.
Yeah. Are these like compounds that are volatile that these
Yeah, yeah,
bees and
yeah,
wasps
they are
could sense?
volatile. Huh,
Right.
they are volatile.
Right. Yeah. Good. So you have more or less answered two options here. You told that
Yeah,
option B, where the flowers mimic the pollinators mate.
yes,
And in order to do that, first it has to elicit or express these volatile compounds that will travel in air and attract
yeah,
mates.
yes,
And once pollination has happened, it produces these repelling compounds as well, so that you don't have other species that would bring in different pollens.
yes.
So, and do you know what phytoalexins are?
No, sir, no, I have no idea about it.
Okay.
Or
These are,
phytoalexins are responsible for the fragrance.
uh, is it so? Are fragrant compounds not called as aromatics?
Mm,
Phytoalexins are secondary metabolites, right?
mm,
But they are produced where you have an attack or an infection in plants. So this is again a stress response, and this would not attract pollinators. So if you think about fragrant
okay,
compounds, that the aim of those fragrant compounds is to attract pollinators.
okay,
Phytoalexins cannot be fragrant because it has already undergone some stress and it wants to reduce the stress. So it is an anti-stress response. So it wouldn't want pollinators. So phytoalexins cannot be fragrant.
okay.
Right.
So it can be called as
Microbial, antimicrobial or
aha,
anti-stress, anti-infection.
anti-stress or
Yeah,
antimicrobial. Okay, okay.
yeah, yeah. So this is just a picture like how it was shown in the recorded lectures where the leaves would be similar to the mating partner of one of these insects. So these insects would come with pollen that would eventually fall into the plant, thereby leading to pollination. The next question is find a statement which is true regarding plant metabolism. Option A, both the primary and specialized metabolism in plants are adaptive with high turnover. Option B, specialized metabolism in plants is indispensable for growth and development, but dispensable for survival. Option C, in both primary and specialized metabolism, high enzyme activities are observed with no feedback regulation. Option D, specialized metabolism in plants is unique and diverse, whereas primary metabolism is universal and conservative. So, whoever wants to try answering this question can raise your hands after a couple of minutes. You can choose a particular option and start with why that could be incorrect. We would go to the right option once we eliminate all the incorrect options. Yeah, Swati, you can start with one incorrect option and tell us why you think that is incorrect.
Oh. Um, the first, shall I tell the true
Incorrect
answer?
option. No, no, we'll go to the right option once we eliminate the wrong options.
Okay. Uh, First incorrect. This one. So the B. Specialized metabolism in plant is indispensable for growth and development, but dispensable for the survival. So it is indispensable for the survival.
Hmm. So again, we'll go back to what specialized metabolism is so that people are aware why it is indispensable for survival. So what is specialized metabolism? Is it the metabolism where you get secondary metabolites?
Yes. Secondary
And
metabolites, the specialized metabolism in plants.
right. And where are secondary metabolites used? Are they used for survival?
Yes.
Right. So that's why you think option B is incorrect. Okay.
Yes.
Do you also want to comment on other options? Other incorrect options?
Uh, this in both primary and secondary specialized metabolism, high enzymatic activities are observed. This one, I think for primary it is high,
Hmm.
and for secondary it is low enzymatic activities. I think so.
True. So, yeah. So, here, high turnover would talk about enzymatic turnover of the enzyme. Enzymatic turnover, right? Now, why would specialized metabolism need low turnover? Any idea about that?
No.
Okay. So, specialized metabolism is at a specific stage. It shouldn't be consuming all the energy. Having a high turnover enzyme would
Huh.
mean that it would consume a lot of energy. That is one of the reasons why evolutionarily these specialized metabolism or secondary metabolism enzymes do not have a high turnover, which is also why people do metabolic engineering, where these secondary metabolite producing or converting enzymes are designed, the, they are engineered to have higher turnover. So you can link it with respect to the energy metabolism.
Okay.
Good. Now, and one more reason you can think of with respect to this turnover is that these enzymes require cofactors. They cannot, in many cases, they require cofactors. Without the presence of specific cofactors, these enzymes are not functional. So, and the cofactor availability can again be linked to the energy metabolism. So, at the end of the day, it's more to do with energy conservation within the cell. That's why these are low turnover enzymes. Right. And so now we have options C and D. So,
In the C also, it is
huh,
low enzymatic activities for secondary specialized metabolism.
right.
Primary, it is high
But
enzymatic
we
activity.
have
So I
covered
think
that.
D is the
So,
answer.
what about feedback regulation? Do you think these enzymes need feedback regulation? Specialized metabolism enzymes? Do you know what feedback regulation is?
Means primary molecule, it will depend on the feedback mechanism for the primary. Is it
Can you repeat? I didn't get what you said.
with no feedback regulation? I'm
No,
not sure.
no. You can first tell us what you know about feedback regulation. We'll take it from there.
Feedback inhibition or feedback regulation means the molecule from where it is starting, the starting molecule, it will be uh, acting or inhibiting the first molecule from where it is synthesized.
So, are you
Feedback
talking
regulation.
about a substrate inhibiting a reaction or a product inhibiting a reaction? So, an enzyme has two components. It takes a substrate, it transforms it into a product. Now,
Hmm.
will the substrate inhibit the reaction or the product inhibit the reaction?
Substrate inhibits the reaction.
If substrate inhibits the reaction, substrates cannot be converted. So, do you think
Hmm.
substrates can inhibit reactions?
No.
Huh. So it's the products that inhibit the reaction. So feedback regulation is a process where once a biotransformation has happened, the product goes and binds to the enzyme's active site or an allosteric site,
Hmm.
thereby sending a message that enough of the product has been produced, so stop producing
Yes.
the enzyme, stop converting the substrate.
Hmm.
So this is needed with respect to secondary metabolism because secondary metabolism is not happening constitutively. It has to start and it has to shut down. So this feedback regulation acts as a mechanism to shut down your secondary metabolism. So feedback regulation exists in secondary metabolism. So with that, we will eliminate option C as well. So option A, B, C are incorrect, and option D is correct. And this specialized metabolism, it's unique to plants, unique among plants also. Whereas primary metabolism, which is needed for production of all the essential compounds for it to grow constitutively, that is universal and it is conserved. So we looked at specific questions on week one's lecture. Now we have a pseudo question session. Like these questions are not specifically on any week's lectures, but rather generally about the course. So again, if it's interactive, it will be easier for you to understand. So the first question is, what do you understand by the term pharmacognosy? And the options are study concerned with uses, effects, and modes of actions of drugs, study concerned with practice or profession of preparing, preserving, compounding, and dispensing medical drugs, study concerned with medicinal drugs obtained from plants, study concerned with laboratory synthesis of natural products. So whoever knows the answer can unmute and start.
Yeah, it's C. Study concerned with medicinal drugs obtained from plants.
Do you
But it
think
should,
that is pharmacognosy? Can you break down the word pharmacognosy?
yeah, study of
Okay, what is pharma?
blind
Sorry.
study of drug.
Yeah. Okay. And cognosy is understanding.
Hmm.
Now, here in pharmacognosy, do you think it's only about plants, or
No,
does it
it's
also
mineral.
include?
No, it includes mineral also. and marine products also, and then
So the source
animals
is not
also.
just plants.
Yeah,
Okay, so that means option C. Do you think it's correct or wrong?
then it's wrong.
Okay, okay. And in pharmacognosy, do we synthesize the products or extract the products?
Yes, sir.
So I think option B will
Yes,
be
we
correct.
complete the we do the extraction process in pharmacognosy.
So we don't do laboratory synthesis, at least to
Isolate.
start with. Yeah. So option
Ha,
D is also wrong.
correct.
Okay. Uh, yeah, Anvay, you wanted to mention something?
Yes, sir, I told that the option B will be correct,
Hmm.
but it's not.
That looks like more of like pharmacist definition.
Yes.
So we can't
It's,
really go with option B.
sir, option A is an correct option,
It,
wherein
hmm,
we find out the therapeutic uses,
hmm,
its mechanism, and what will be the use. So I think C is also correct actually. It's study concerned with medicinal drugs
hmm,
obtained from plants, and then whatever is there, its uses, effects, and mode of action. Because
hmm,
these we cannot dispense the medicine
hmm,
when it is made, and we cannot synthesize in the laboratory the natural products.
hmm.
So I think A and C.
So I would say here multiple options seem incorrect and correct at the same time. You just need to
Okay.
specify. You just need to
uh,
specify uh, a few more, a little extra information. Like for example, if you say option C is correct, you need to mention that it's not just from plants, but from other
other.
sources as well. And to
So
me,
then I go
that
for
would be great.
answer A.
Answer
Answer
A.
A then.
But answer
Yes.
A talks about drugs and they do not, it doesn't talk about natural compounds. It doesn't say that drugs are natural
Okay.
compounds and the source is not specified. So C is the closest to the right answer, I would say.
Right
But again, it's
answer.
subjective.
Okay.
Yeah. Anyways, this is not very technical, so you need not be bothered about questions like these. And yeah, now, which does not come under the umbrella of metabolic engineering? To enhance the turnover of a desired metabolic pathway, to introduce a desired metabolic pathway in a heterologous system, to chemically modify plant specialized metabolites, to reduce the turnover of non desirable metabolic pathway. So, yeah, whoever knows the wrong answer, whoever knows the answer can first justify why a particular option could be incorrect. And then we'll reach the right answer.
So A, B, and D are incorrect, sir.
A, B, and D are incorrect.
Oh, so that means they are the answer.
Huh.
Because metabolic engineering is something where you actually enhance the turnover of a desired metabolic pathway that is required.
Right,
So to increase the yield of the any metabolite that you're targeting.
right.
And second, it can also be introduction of a desired metabolic pathway in a heterologous system where we might see that many biosynthetic pathway that is seen in plants are engineered in unicellular eukaryotes like yeast and many other vice versa here and there. So that is also comes under metabolic engineering.
Yeah, but
And
just a minute. Yeah, what you said was right. In addition to what you said, here heterologous system means this system cannot naturally produce that compound. So
yeah,
if you put a pathway from plant in E. coli, a microbe,
yes,
or yeast, then
yeah,
that pathway is heterologous to the organism. It
yes,
is
yes,
not native. Huh.
yeah.
Yeah, continue.
Okay. And D, it is to reduce the turnover rate of non-desirable metabolic pathway. So because uh, when you introduce say in metabolic engineering basically, so you have many aspects that you have to look into. So if
Hmm.
you want a particular metabolite to be produced in higher quantity, you will
Hmm.
have to shut down other pathways,
Hmm.
or say reduce the flux towards those pathways, so that the pathway that you are targeting is enhanced much. So this turn off of non-desirable pathway can also be an option, like it comes under metabolic engineering.
Right.
But chemically modifying a plant specialized metabolite is something that's not, like, that can't be done using metabolic engineering. So that's a different aspect of chemistry where you design drug and whatnot.
Right. Yeah. With respect to option D, here one can imagine that when you have a pathway within a cell, you have multiple metabolites, and the metabolites usually get converted from one to another. They rarely accumulate within the cell. So, this means that there are multiple competing pathways that are interested in transforming a particular precursor. But if we shut down those competing pathways, that is what is mentioned here as reduce the turnover of a non-desirable metabolic pathway. And like you mentioned, to chemically modify plant specialized metabolites, we have to extract the metabolite and do it in vitro, which is outside the cell within glass, outside the cell. So that wouldn't come under metabolic engineering, but rather as chemically modifying compounds or drug designing or yeah. The next question is, among the following, which is not a function of plant specialized metabolism? So option A, to induce flowering. Option B, interact with the biotic component of the environment. Option C, help the plants to survive in competitive environment. And option D, synthesis of aromatic amino acids in plants. Yeah, so whoever knows the answer uh, can unmute and start by mentioning why an option you think is incorrect. Did someone raise their hand?
The
Yeah, Nisha.
correct answer,
You can start with
correct answer,
an incorrect option. Why you think a particular option is incorrect?
incorrect.
Hmm.
Uh, is not a function of plant specialized thing. Interact with biotic component of the environment. So, secondary metabolites,
Hmm.
they are produced under some stress to as a defense mechanism of a plant.
Hmm.
So, if a some environmental stress is there,
Hmm.
obviously the secondary metabolites will be produced.
Right.
So, this has to be the correct one. Interact with biotic component of the environment. Same with the C, that is helps the plants to survive in competitive environment.
Environment. Right.
Yeah, this is also correct.
Right.
And then it helps the plants to induce a flowering because of some that no, not exactly, but the answer should be D, I think so, because this is about the primary metabolism,
Good.
not the
But
specialized metabolism.
uh, yeah. So amino acids are required throughout the cell to
Yeah,
synthesize proteins. So you
yes,
can't eliminate that also.
yes.
So that
Mm,
is the that is something that does not require specialized metabolism, but rather primary metabolism. So that is the right answer.
yes, sir.
Okay. Now we have a specific question. So here the question is, which one of the following represents specialized metabolic pathway? Option A, beta oxidation of fatty acids. Option B, pentose phosphate pathway. Option C, Halliwell-Asada pathway. And option D, mevalonic acid pathway. So before we go to the right answer, So to think about this question, think about pathways that you studied across species, plants, animals. So that would tell you what a primary metabolism pathway is and how it is different from specialized metabolic pathway. Yeah, Sushal.
Hello.
Yeah, I can hear
Yeah.
you.
So I think the answer is D. Mavulonic acid pathway is the specialized metabolic pathway among all this.
Do you know what mevalonic acid pathway is?
Yeah, it produces the precursors, right? IPP and DMAPP, which are like the building blocks for like many plant terpenoid synthesis. Or I mean, need not be plant terpenoid. It can like there are many things that come out of mavulonic acid pathway. So and all those are specialized and are particular to different plants based on whatever enzymes they have after the production of IPP and DMAPP.
But okay, doesn't mevalonic acid pathway produce fatty acids, cholesterol, and all of
Yeah,
that?
it produces. Yeah, I mean, after mavulonic pathway, whatever enzyme, say farnesyl pyrophosphate, and then
Hmm.
after that you have many other
Guanine.
enzymes, and so that after say particular part of mavulonic acid pathway, so any enzymes you give differently will give that different metabolite is what I have studied. So with that is one thing I am guessing because so each if you give farnesyl pyrophosphate that comes out of mavulonic pathway
Hmm.
as a precursor to different plants or animals, so they're able to produce different compounds based on whatever enzymes they have after the following enzymes after FPP.
Yeah, so okay, that is fine. But is
Yeah,
FPP essential for the cell?
is FPP?
Essential for the cell
Yeah, it is very essential.
always,
Yeah,
independent of the condition.
yeah.
So wouldn't that make it a primary metabolite then?
uh Yeah, but I that's what I don't know how far the mavulonic acid pathway can go, and what is the last um, compound that you tell consider is from the mavulonic acid pathway.
No. So it's it's very simple. As long as you know that you have a particular precursor that is present that gives rise to an essential metabolite, you need to
Hmm.
call that pathway as an essential pathway or a primary metabolic pathway.
Okay.
So that way, I wouldn't consider mevalonic acid pathway as a specialized metabolic pathway, but rather I would consider it as a uh, primary metabolite because, as far as
Okay.
I know, it gives rise to these geranyl compounds and farnesyl compounds, which
Yeah.
are in a way related to cholesterol as well. So, and because
Okay.
cholesterol is essential to the cell, you can't call it specialized. And one more way to think about this is like how I mentioned earlier to see whether these pathways are present in different organisms or different kingdoms. Mevalonic acid pathway is present across eukaryotes. Like the same is with pentose phosphate pathway and beta oxidation. But you don't see that in the Halliwell Asada pathway. So that is a stress response pathway when you have different stresses. And these are oxidative
That's
stresses and
used
how
for
the plant.
difference, right?
Sorry?
Difference. This pathway
Yeah,
is used. Okay.
yeah. Yeah. So a plant expresses or experiences a stress, and in defense, it does something. So this pathway comes under this specialized metabolism where it acts in response to different stimuli that I mentioned here. So In that sense, I would say Halliwell-Asada pathway is a specialized metabolic pathway and not mevalonic acid pathway. Um, the next question is identify the correct chemical skeleton from the following that plants use for the synthesis of phenolic compounds. So, yeah, this is again a simpler question to answer based on chemistry. So, whoever knows the structure, you can start by mentioning what each structure here is called as chemically. Yeah, does anyone want to try this? Structures of compounds, basic chemical compounds? Yeah, Sushal. We start with the incorrect options like we usually do.
Okay.
Yeah,
So the D is cyclohexane.
okay.
So that doesn't have any. It's not a basic skeleton for synthesis of phenolic compounds because phenolic compounds are usually aromatic.
Yes, they are aromatic and they are unsaturated.
Yeah, unsaturated.
Yeah.
And C again, on the other hand, it's glucose,
Hmm.
and even that has no role in synthesis of phenolic compounds. Like again, say it should be aromatic and unsaturated.
Okay. Okay. I would add on to something once you finish. Right.
Yeah.
Huh.
But B, it's B is what it's pyramid. What is it? I exactly don't know what is B.
Purine, pyrimidine. You can choose. So one of
Then it's
these.
purine. Purine.
Okay. Yeah. So purine has two rings.
Yeah,
Pyrimidine has one ring.
one ring. Huh.
Yeah.
So again, yeah, purines are not used in synthesis of phenolic compounds.
Hmm.
Yeah, because there's no. It's not a. The phenolic compounds are not having. They doesn't have a heterocyclic aromatic ring. So it's
Yeah. Usually phenolic compounds lack nitrogen. They are usually
yeah.
carbon, hydrogen, and oxygen.
Yes. Huh.
Right.
So and yeah, it ends up with A as the answer, where it it matches. Say it is aromatic and it's unsaturated, and you have alcohol groups also. You can build on it and make
Right.
varieties.
Right. Just another additional information. So, what we see here as option C, these monomers of carbohydrates. So, these have more solubility in water. In plants, these phenolic compounds, such as flavonoids, are hydrophobic. So, they're not water soluble and they're not bioavailable. So, what plants do when they want to transfer or improve the solubility of specific flavonoids, or not just flavonoids, other phenolics, is that they do a process called as glycosylation, where an UDP glucose is taken as a substrate, and glucose from the UDP glucose is attached to the phenolic or flavonoid moiety. So you have these glycosylation processes that is also helping you to improve the bioavailability and solubility of these phenolic compounds. But these are not a chemical skeleton. So the right option is option A. But I just wanted you guys to be aware that option C is also a part of the phenolic uh, compound in some cases. The next question is identify the specialized metabolites from the following compounds. Option A, fructose 6 phosphate. Option B, lignin. Option C, triacylglycerol. And option D, selenocysteine. Yes, Sushant.
So fructose 6 phosphate, yeah, it's there in glycolysis and it's found in all plants
Right.
and bacteria, animals, whatnot. It's universal. Triacylglycerol is also, uh, say, it's required for the fatty acid
Hmm.
synthesis. And selenocysteine, as you know, is it's an amino acid,
Hmm.
but that's seen in, say, not all organism, but it's a basic essential amino acid, so you can consider that as a metabolite.
Wait. Uh, with respect to amino acids, when you call something as essential, do
Yeah.
you mean that the cell produces it or the cell consumes it from out other sources?
Uh, excuse, I did not
Okay,
understand.
so amino acids are classified as essential and non-essential.
Yeah, yeah.
Essential amino acids are those that have to be consumed from outside,
Yes.
which means that cells cannot produce that.
Huh.
So, do you mean to say that selenocysteine cannot be produced by cells?
No, no, it can be produced.
Okay,
It can be produced.
okay,
Yeah, but it's not a secondary metabolite, is what I am telling.
okay. And lignin?
Lignin, I can, yeah. So my answer is lignin. So lignin is a special metabolite because lignin is seen in not all plants, but only few plant species that have, say, definite function where they have to make the shell hard and seeds coating hard, whatnot, bark, and many other place.
Okay, okay. So, one way to look at this again would be to look at the structure of lignin. So, earlier we had seen that lignin forms by, so this is the process where you have paracameral alcohol, coniferal alcohol, and synapyl alcohol, which oxidatively couple to form your lignin. Now, if you look at this, all of these are not primary metabolites. So, with that, you can say that lignin is the specialized metabolite among the options given here. And one thing of interest is that selenocysteine is not naturally observed in plants. There are very few cases where plants have proteins with selenocysteine incorporated in them. The first reason being there is the incorporation of these amino acids depends on the codon, the three-letter nucleotide, which can be recognized by a tRNA. So the availability of these codons and tRNA that complementarily binds to these codons is a limitation. Second is the presence of selenium itself. So there is one paper that was recently, uh, not recently, sorry. There is this one paper that I came across which confirmed that there is a specific codon that could uh, introduce selenocysteine and it's present in a particular enzyme from Chlamydomonas. So if you're interested, you can look at this paper. In general, we use selenomethionine when we want to use that as a probe to be able to study other parts in a particular process, like for example, X-ray crystallography or so. The next question is, Identify the compound which is not used by plants to attract pollinators. So here, if you look at this, pollinators get attracted to pigments that are the colors that are available on plants, on flowers, nectar, which is the sweet that plants offer in order to attract the pollinators, and volatiles. These are again the aromatics which have a particular fragrance for which pollinators come. Whereas starch is not usually provided because sorry, because starch is the carbon source for plants. Name the most suitable technique for separating metabolites from the following: electrophoresis, chromatography, microscopy, and spectrometry. Yeah. Does anyone want to answer this question? Yeah,
It's
Nisha.
chromatography. It's chromatography.
Okay. How is chromatography different from other options?
Microscopy, though it will be used to visualize the cells.
Okay. To zoom in and see. Okay.
Hmm.
Ah. And what about spectrometry and electrophoresis?
Electro. That's basically electrophoresis. It is used for the separation of like proteins and your RNA, DNA molecules, not for the secondary metabolites.
Okay. So for polymers, whereas
Yeah,
secondary metabolites are monomers and they are small.
yeah.
Okay. And spectrometry? It's actually spectrophotometry.
Spectrophotometry. It's not for by spectrophotometry we cannot separate. I think
It is an identification technique.
identification technique. Yeah.
Right.
Yeah.
So these
Uh-huh.
molecules will have specific bonds that can
Mm.
absorb specific wavelengths of light. Could
Yes.
be either visible light or infrared, could be any wavelength, but these absorb and you see a difference in the absorption with respect to specific functional groups. So spectrometry or spectrophotometry is used for
Correct.
identification of functional
Identification.
groups. Right. The last question is identify the plant-based drug from the following. And we saw a question related to this here. So it's quinine here. That is,
Quinine.
Quinine.
Yeah.
yeah, yeah,
Quinine.
yeah. So with this, we come to the end of today's class. If there are any questions, you can ask me. Sorry for the delay. It's we are going beyond time. Sorry for that. But if there are any questions, I'd be happy to answer. Or we can end the class here. Okay, if there are no more questions, I'll end the class. Thank you for attending. So the next class we'll have, yeah, Deepali, sorry. Did you want to say something, Deepali?
No, no, sir. I would like to say you thank you. Nothing
Okay,
else.
okay, thanks. Right. So we'll have the next class again on Monday, 6 to 8 next week in the same Google Meet link. Thank you.
Okay, sir.
Thank you.
Thank you.
Thank you, sir.
Thank you.